morning, everybody. My name is Andreas Mayans. I'm honored to chair this first session today, and I welcome you all to this second day of this interesting meeting. So I have the pleasure to introduce to you Dr. Julia Garcia-Pardo, I hope I pronounced it fine, from the XIII AIDS Research Institute in Can Ruti in Badalona. Julia, before she will go talk about how HIV is sort of avoiding to be eliminated by the immune system, something we heard yesterday about how pancreas tumors do that, and now today we hear about how HIV does the same thing, avoiding immune destruction. And before she is going to talk about this, then let me just introduce her a little bit. She is from here. She studied biochemistry and had a degree there and made her PhD in immunology at the Autonoma University in Barcelona in 2005. Then went to Oxford to Philip Golders' lab with the Marie Curie Fellowship. She came back here to Barcelona with the Miguel Servet Fellowship as a Miguel Servet researcher. Then since 2013, she has her own research group at XIII. And as the title says, like she's working on, and the group's name, she has her own group, Viral Immune Evasion and Vaccines, so you easily understand what she is doing. But besides of doing research, she has another obligation or pleasure to do. She's deputy director of the IGTP at the Research Institute of Can Ruti, and she's trying to combine these two activities with quite some success. So I let you now talk, Julia. Please enjoy the talk.
Thank you. Good morning, everyone, and welcome to the second day of your conference. First of all, I would like to thank the Catalan Society of Immunology for inviting me to share some of our perspective in the HIV research field. I believe that the topic of this year, immune evasion of pathogens and tumors, it actually sets the perfect scenario to talk about HIV. I would like to think about virus as the artists of immune evasions in nature. In fact, these are tiny microorganisms that they are infectious, and they have co-evolved with humans and animals through millions of years. Thus, viral replication is based on the efficient recruitment of cellular functions and is basically due to interactions between cell proteins and viral proteins. As a consequence, during this long evolutionary race between the human immune system and the virus, humans, we have specialized to control pathogens through immunity, and viruses have specialized actually to counteract this immunity, favoring in this battle by this rapid adaptation to environmental changes. So this is HIV, this is one of the viruses, and I will going to bring you a brief overview today about first the basic virology, some key concepts that I think are very important to understand how HIV can immunovate human immunity, and also some mechanisms that has been long-term described for HIV immunization to set probably the challenges that we are facing today, trying to cure HIV. I think you have some talks yesterday by Josep Maria Gatella and Felipe Garcia, and they give you a little bit of overview what we are facing now in the field of HIV, trying to cure the disease. And finally, I will give you actually nowadays some parallelisms that we are, and some challenges that we are facing between HIV cure and tumors. So this is an HIV particle. An HIV particle is spheric and is characterized by a big-capped lipid membrane that actually has, I think I lost the pointer. It's not here. Okay. It's okay. I was hiding on the back of my computer. So it has some GP160 proteins in the membrane of the virus that basically are the ones that lead the virus to the entrance of the infection of the cell. And if we peel this virus as an orange, we can get a second layer that is defined by the matrix capsid and is part of the protection of the inner proteins. And probably the most important bit of the virus, so one of the most important bits of the virus, is this electron-dense core formed by the P24 capsid proteins that protects two single molecules of RNA that constitute and form the HIV genome, together with other viral proteins, including the reverse transcriptase and the integrase, that are crucial for the integration of the genome in the host cell. So if we think about how virus is infecting its primary targets, there are CD4s. So if we think about HIV, HIV is actually causing an immunity deficiency. And it basically enters through CD4 receptor and CCX4 or CRC5 co-receptor in the cell. And there is two important features that we need to bear in mind. One is that HIV needs to retrotranscribe and produce two cDNAs in order to integrate the host genome in the viral genome into the host cell. That makes that the cells will keep, we keep HIV genome forever in our cells. And the second one is the retrotranscription that helps to RNA viruses to have such a big variability that we are going to discuss in a while. So, although today we can block HIV replication with a huge battery of combined antiretroviral treatments, and currently we have around 80 drugs approved by the FDA for HIV treatment, and we also know that we can measure antiviral responses against virus in our infected individuals, either adaptive T-cell responses, we can measure antibodies, we can measure innate immunity during the natural course of infection. Of course, we can measure and we can identify many restriction factors, intracellular restriction factors that are able to block HIV replication at different stages of the cycle of replication. Despite all of that, HIV has been able to spread around the world. And currently, maybe all of you think about HIV, something that is chronic, something that is not happening anymore, and we have this view that is solved because we have a very good antiretroviral treatment. And in fact, this is not the case. HIV has a lot of society problems that are involved. It bears a lot of stigma and discrimination, but behind that, we need to face what is the pandemic today. So we have around 38 million people infected worldwide. More than 60% are located in sub-Saharan African countries, and we have around 1 million deaths per AIDS a year. And also, we are continuing getting new infections at a rate of 1.8 million per year. So it's quite unsustainable to keep 2 new millions infections a year in terms of how we can provide antiretroviral treatment and how this could be sustainable for treatments that are needed for life. So why HIV has been so successful, I think, as I mentioned before, is due to their characteristics as a RNA virus. And I will going to try to go through 3 different mechanisms of HIV immunization that I think bear part of this power of HIV to spread around the hosts. So one of the mechanisms, of course, is how virus can generate viral proteins that are really specialized to counteract immunity and to counteract restriction of the cell. How HIV variability, in this case linked to this nature of an RNA virus, can favor viral immune escape, and I will focus in T-cell responses because that was part of the work we are doing in the lab. And also, of course, how HIV is able to persist in these immunocells, mainly resting CD4 T cells, and maintain transcriptionally inactive in these cells. So on the first mechanism, I'm not going to talk a lot in detail about this, but basically we know that there is certain viral proteins such as EpoVec3G, SAMSD1, TRIM5, and Teturin that are known to be able to counteract HIV replication at different stages of their cycle of replication. So by hypermutating the virus, by counteracting the pull of DNTPs in the cell, by blocking and maintaining the virus attaching the membrane, or by allowing the premature capsid encoding of the virus, all these intracellular proteins can block HIV replication. However, we know also that the viral genome encodes a specific proteins such as BIF, BPX, BPU, or can recruit host cellular proteins such as cyclophilin to avoid the control by these restriction factors. So a second mechanism of immunoevasion, as I mentioned before, is linked to the RNA nature of HIV viruses. And it's always nice, and I think this is some slide that we all use to try to explain how RNA virus is, which is, that you get accounted how variables are RNA virus. So if you look at this picture, you can see here, this is a phylogenetic tree that basically identify how viral strains vary across, in this case, the wall in an annual infection by influenza. And as the tree branch and spreads the branches, we can see an increase in variability. So, in fact, the annual variability of influenza is similar of the variability of HIV in a single individual. So if you think about, we need to generate a new vaccine for influenza every year. For a single individual, we will need to we can tell like that, generate many vaccines for a single individual every year, because of course the virus is evolving very quickly within an individual. And even more, if you think about just one of the places that we can see more variability of HIV across different subtypes, that is Central Africa, you can see how complex could be tackled so much variability of this virus. And this is also a picture that represents HIV subtype diversity, and this is Central Africa, so that would be the equivalent of this phylogenetic tree, and you can see that each color represents a different subtypes. So you can see how how HIV variability is actually very important for potential immune escape. It might give an advantage for immune escape, but it also is a really major handicap what we need to face when we try to design a new vaccine or design vaccines against HIV. So how HIV is so genetically variable? If we think about numbers, I think one of the key facts about HIV is that it has a reverse transcriptase. This reverse transcriptase is an enzyme that lacks of proofreading activity. So in fact, it's not accurately transcribing these two RNA single-strand molecules of the HIV genome, but in fact, it's introducing changes every time that it's transcribing. Moreover, the viral production a day is around 10 to the 9th and 10 to the 10th virions, and we have more than 10 million replication cycles a day. This basically results that we have a probability between 0.1 and 1 mutations per genome and per replication cycle. And this is called a quasi-species structure. So actually this name was coined by Esteban Domingo in the 1978, and that basically described the behavior of many RNA viruses that are composed by a swarm of very related genomes, but with a high diversity. And this high diversity basically provides an advantage to that against environmental changes. And of course, every time that we generate an immunoresponses, we are facing the virus is facing an environmental change. So this is a brief overview of the natural history of HIV. So when an individual gets infected, we have a peak of viral replication that basically goes to a time of a steady state when virus and hosts are facing an equilibrium between the level of viral control and immunity that basically breaks down at the end of the infection, when actually the patients, they don't die for HIV, but they die for the immunodeficiency caused by HIV. And the viral load basically is going down because it's coming up again, and the CD4 basically completely disappears, and normally immunity kind of fades out also. We're going to focus on CD8 T-cell responses because that's, as I mentioned before, part of the work that we are doing. And it's important to understand, as we face this great genetic diversity of HIV, that every time we generate an immunoresponses, in this case CD8 T-cell responses, we are able to partially control HIV replication. But there is some point that the virus kind of breaks this equilibrium, rising a new viral strain that is resistant or is escaping these immunoresponses, and this is basically happen along the time of this steady state process. So the virus is evolving and racing against the host CD8 T-cell responses. And this is key for how HIV can adapt very quickly to immunity. It has been also described that one of the major driving forces of HIV adaptation is the locus, the chromosome 6, at the locus of the HLA class I molecules. And we know that in this balance between HIV and AIDS, we can set the states, and it has been many times now identified that we have some HLA class I haplotypes that are associated with a slow progression of HIV and control. This is the viral log. So these individuals have significantly lower viral loads, yes, by carrying these alleles. And other alleles that are associated with fast and rapid progression. So that's basically telling us that the HLA class I immunopressure, mainly mediated by CD8 T-cell responses, is actually imprinting signals in the virus because the virus needs to evolve and then the virus needs to escape. And how it does that? So this is really well known by all of you, so I'm not going to get into detail, but we know that viral antigens of infected cells are presented in the context of MHC class I molecules, and this epitope presentation of the protein of the virus can be recognized by HIV-specific CD8 T cells, and this recognition basically leads to the activation of the CD8 T cells and the secretion of cytotoxic cytokines and elimination of the infected cells. This is just the representation of MHC class I molecule. These are the binding pocket of one of the HIV peptides. Actually, this peptide is restricted by one of the protective alleles, that is HLA-V5703. And one of the things that HIV can do is basically to change the nucleotide sequence in one of these amino acid residues, changing in this case from alanine to glycine, and lowering the affinity of the peptide for the MHC class complex, that therefore this new viral peptide that is going to be produced by the infected cell is not going to be recognized. So also we can find here similarities of some of the neoantigens of the tumors. So tumors generate neoantigens to avoid MHC class I presentation. So how we can check this in the lab? So we have our virus, and we can modify this virus by introducing single mutations. In this case, this could be the epitope that I just showed you before. and infect with a reporter virus that produces GFP, CD4 T cells. So this would be kind of when we have only HIV. This is the wild type. This is our mutant. And when we add CD8 T cell clones specific for that epitope, we can find, or we can observe how wild-type viruses are completely abrogated for replication. Meanwhile, mutant virus is able to continue replication. So, you know, single chain can actually alter the pattern of recognition. And when these responses are really immunodominant, sometimes a single chain can precipitate disease progression. And this is one of these cases. So how immunoscape can impact disease control. So this is what we call a natural HIV controller. That's people that is able to control HIV without treatment. Of course, we don't have that anymore because we treat everyone that get infected with HIV, and these are historical cases. But in this case, this individual was able to control for more than 25 years HIV, and this is not the standard. But suddenly, about 27 years, we can observe how the viral load is going up. The viral load is going up. And the CD8 counts are going up, and the CD4s are going down. So there is some point, a breaking point in this pathogen-host equilibrium that basically is associated with this loss of control. This patient has one of these protective HLA alleles that we have mentioned before, that is B1401. And this B1401 response is focusing, as far as is described, in two main epitopes of HIV. One, the EL9, that is located in the envelope of the HIV virus, and the DA9 that is located in a fraction of the capsid protein. So as you can see here, associated with this loss and control, we can see how there is a loss of response, of T-cell response, measured by interferon gamma ELISpot assay, and that is never recovered and is lost over time. This GAG epitope, this GAG responsor, is never present. And also what we observe genetically is that in this epitope, we can see how the virus over time is accumulating mutations and how this mutation here is never recognized by the host. So again, you know, a single chain in an epitope is actually altering how the outcome of the disease. And in this case, someone that has been controlling HIV for very long lost escape associated with the acquisition of a mutation. Also, we have been interested over the last year to try to understand how immunoscape is impacting not only control, but maybe other phenotypes in HIV disease outcome, for example, rapid progression. And this observation comes through a really kind of unexpected observation that we were able to identify many individuals that they progress really quickly to HIV. So in less than 2 years, they have less than 200 CD4 counts. That is very low. And we were surprised because we have few rapid progressors that they have these protective HLA molecules. So we thought, okay, how is that? If you have a good HLA, why your progression is bad? And what we found when we analyzed the virus is that in these epitopes, in these responses by B5801, they are very immunodominant against a TW10 epitope that drives escape towards this mutation at position 3 of the epitope, and all these 3 rapid progressors, they have this mutation at the time probably of infection. And this is another case. This is a B27 individual that again is a very protective allele for HIV, and this is a very immunodominant response against KK10, one of the regions of the virus. So if we look here, one of our rapid progressors, it actually has 2 mutations at the binding site of the epitope in the HLA molecule, but none of the standard progressors that we evaluate. So basically, we did analysis of a cohort of about 25 individuals with Robin progressor. And what we have observed is that most of these individuals, they have an increase in common alleles, and also they have an increase of mutations at the CTL sites, at the epitopes that the CD8 T-cell response is going to recognize. So our hypothesis is that these individuals are getting viruses that are very adapted of their HLAs, and these probably are the ones that are circulating in our population, and therefore, once you get this virus that is very adapted, together with other features that I didn't mention, but this the high replication and the high spectrum of infectivity of different cell types, we get this phenotype. So as a small summary of this part, we can say that multiple mechanisms of immune evasion coexist in HIV infection, that HIV variability account for a very great proportion of the adaptability of the HLA class I derived immunopressure, and immune evasion can modulate disease outcome in natural HIV infection. And of course, all these mechanisms are really important to understand in order to design new therapeutic approaches, such as small molecules, gene therapy, and many other approaches that have been used and we are developing in the field. And it's also very key to inform a rational design of vaccines. So how we can block the virus if the virus is able to evade so many T-cell responses, if we think about in a T-cell vaccine. And I think it was, kind of important to talk about HIV persistence. That is what is driving the field forward nowadays, because as I mentioned at the beginning, our patients are well. We have very potent treatments, but we have newly infections worldwide each year. And of course, we think it's not sustainable to keep treatment for life. So we need to face HIV persistence and to try to eliminate this persistence in order to cure, or at least to find a long-term remission of HIV infection. So how HIV is able to persist? HIV is actually, if you think of a virus that is causing an immunodeficiency, it's actually from the beginning affecting immunity, because it is targeting CD4 T cells. And it is, affecting right from the beginning how our immune system is functioning. So actually, it's working against immunity right from the beginning, on top of these all other mechanisms that we have discussed about. So it is known that HIV persists mainly in resting memory CD4 T cells through different and complementary and simultaneous mechanisms. So one is how cells that carry an aprovirus can eventually get activated and produce some virus that can perpetuate these cells. The other is the fact that this resting memory CD4 T cell has a long-term survival, so they can last for very long without any changes. So, and the third one is how these cells are susceptible to homeostatic proliferation events. So every time that they found maybe not HIV antigen, but another antigen that is around, they would be able to divide, proliferate, and perpetuate itself across the body. This is a picture just to show, of course, we want to cure HIV and eliminate persistency through different mechanisms. One would be limit the reservoir formation, so where the virus is hiding or where the virus is getting at the beginning through different antiretroviral treatments, but of course we also want to reduce the reservoir at the end to see if we will be able to have a sustainable remission or a cure of HIV. That could be done by maybe render HIV cell resistant to infection. So by using gene therapy approaches and depleting, for example, CCR5, that is essential for the viral entry, but is not essential for the human life. How we can enhance immunoresponses through vaccine and other immunotherapies, such as antibodies, and how we can remove this reservoir, let's say just reactivate its cells and allow for the reactivated cells being eliminated by immunity. And this is basically is changing how we approach HIV in many different aspects and all at the same time. So, you know, we have, the field is developing broadly neutralizing antibodies. Vaccine is facing the use of immunocheck blockers in HIV infection, latency reversing agents to flush the virus out, gene therapy, and even hematopoietic stem cell transplantations in very rare and exceptional cases. So I'm just going to show you a case that basically composed 3 of these elements, because we identified one patient that had metastatic melanoma and had HIV at the same time, and he got treatment with one of these immunocheckpoints inhibitor, a PD-1 inhibitor. And basically, the theory in the HIV, one of the proposals is that CD4 resting T cells, they express, or they overexpress some of these immunocheckpoint receptors, such as PD-1. So if you are blocking PD-1, we hope that we can reactivate these cells, inducing the shock and the production of viral particles. And at the same time, if we can target exhausted CD8 T cells that upregulate inhibitory receptors, such as PD-1 and others, we would be able to regain HIV-specific CD8 T cell function in order to eliminate the reactivated cells. And this is some of our data. That's basically when the patients get infusion of anti-PD-1 every 3 weeks, we can observe an activation of CD8 T cells, very robust activation and sustained in many of the different CD8 T cell subsets, but mainly in effector and effector memory T cells over the 9 weeks of treatment, and then it kind of goes down to basal levels. So PD-1 is altering the activation of these CD8 T cells. It's also transiently increasing the HIV-specific CD8 T cell responses, mainly with the TNF phenotype. And we see then kind of an increase over time of HIV-specific cells producing IL-2 and interferon gamma, but kind of a delay. And it also has some effects on the HIV-specific serial T cell responses. So we were hoping, because these patients have a very good immunity and conserve, to see some effect of the reservoir, but no. So this is the parameters of proviral HIV DNA. So what is the amount of virus that is carrying the cells of these individuals? And we can see a transient decrease at week 6 and at week 27, yeah, and 3. But not an overall reduction of the HIV reservoir. Of course, this is opening a lot of questions. So why we cannot see these effects? There are some patients that they could be more susceptible to these molecules or not. There is only one report case that has shown a decrease in the reservoir by the use of anti-PD-1. So I think this is kind of an exciting new field that many groups are pursuing now. And on top of that, HIV is hiding, but HIV is hiding in many places, and it's hiding in a lot of different cells around the body, and in a lot of immunocells. So it's hiding in CD4s, it's hiding in alveolar macrophages in the lung, is hiding in thymocytes, is hiding even in the brain in some macrophages. So to tackle HIV cure, also we need to face this amount of complexity of the overall system and organic system. And then this is just basically the last slide that is also nice, also taking into account the topic of the talk and the conference, how immunoevasion is important in the different fields pathogens and tumors. And basically what we are kind of facing nowadays is somehow these fields are converging. Somehow we are realizing that we have common challenges. And if we think, for example, about the tumor microenvironment and a lymphocyte node, where we know the HIV can hide and is less, and CD8 T cells, for example, are not allowed to enter or not in a big frequencies. We need to face this complexity of microenvironments within the tumor or the lymph nodes in the case of HIV infection, including this proper kind of metabolic microenvironment. This is immune exclusion that we are facing. We are not getting there. And how we can manage to get there? Do we want to get there? How this is transcriptionally regulated and how we can modulate by many strategies that might be common facing HIV cure and facing tumor remission. And this is my last slide. I would like to thank all the members of the team. I'm looking for a postdoc, so if anyone is willing to join us, is welcome. And all our collaborators, of course, the patient and the funding agencies that support our work. And thank you all for your time.